All right. Looks like I'm up and running here. Thanks, folks, for tuning in from the internet. I'm sorry a lot of you had your hopes up for coming in and doing all this stuff in person, but, got to be responsible. And we went ahead and moved the actual DNA class to April 23rd. That's about 5 weeks from now. And hopefully by that point, we can get everybody in here and show you hands-on. But in the meanwhile, Alan and I have got the entire process set up for you, and we're going to show you from start to finish how to do PCR on mushroom samples. And so just a quick primer on what PCR is. So that means polymerase chain reaction, and it's a way of taking a organism's DNA, chopping a little piece off of it, and then copying that little piece billions of times. And then that little piece of DNA, once it's been copied billions and billions of times, we can send it off to be sequenced. And so the sequencing process is where it basically gets tagged with some fluorescent dyes, and each dye attaches itself to a different letter of the DNA alphabet, the A's, the T's, the C's, and the G's. And then a laser will scan over that, and if a specific one fluoresces, it captures that, and then you get a chromatogram as the result, and that's going to be kind of showing you the individual letters, and we'll go into that a little bit later. So the method that we're using is called extract and amp, and here I'm going to show you the formula for that. I'm going to go ahead and put this up on the internet, but if you want to copy this down right now, this is the recipe if you want to make it for yourself. Now, extract and amp is a commercial product. We've just basically taken that and made it open source, and somebody did. We didn't actually do that, but it's available, I think, through the Wikipedia page of Counterculture Labs, and then I'll make sure that there's a link to it on this YouTube channel and on my Facebook and Alan's Facebook. Speaking of which, Alan is going to do the stream from his Facebook as well. So I'm actually doing it from my Chromebook. You'd have to have 1,000 subscribers to do it straight from a phone. So if you want a better quality streaming experience, go check out Alan Rockefeller's Facebook page, and we'll have that up on there. So the other thing I should mention is that all the reagents that we're using here came from a place called the Odin. And so it's going to be theodin.com, the-odin.com. They are a biohacker supply website. They're based here in Oakland, California. And if you want to do any of the stuff that we're doing today at home, you can purchase pretty much everything you need. Of course, you can't buy the mushrooms there. You'll have to find those yourself. But everything else that you need, you can find on the Odin, the-odin.com. They've supplied us with the primers that we need, the master mix that we need, a DNA ladder, which is going to show us how big our piece of DNA is. And you can also buy the thermal cycler, which is the device that's going to do the changing of temperature required for PCR. And so they sell one just like this called Pro PCR. And so I have one of these. And then they also sell an even more smaller portable unit, which the name escapes me, but go ahead and check out the website. It's going to be on there. And that one is called like the pocket PCR machine. You can actually stick that in your pocket and take it with you on a hike. And I guess if you want to sit down for 3 hours and drink a beer while you do PCR in the field, you can do that. And so again, everything you can find to do this is on the-odin.com. So without further ado, I'm going to go check out what Alan is up to right now. All right, so this is Alan Rockefeller. He's going to be doing the grunt work here, the pipetting and the setting up for PCR. So why don't you tell me about what you have right there, Alan?
So we got 4 different mushrooms that we're going to barcode today. This one's really cool. It's Microglossum viridae, but there's actually a couple Microglossum viridae in California. So who knows if either of them are the real Microglossum viridae. So that's one that I've been wanting to get sequences of. And then Gluteinoglossum is this cool little black earth tongue. And this one, it's like a Gluteinoglossum, but it's got a slimy stem. So this one is pretty interesting. Gluteinoglossum is a genus that was recently made. Then we have a mycotic spore mat. So this is pretty mysterious because it never really makes mushrooms. It's just this white mycelium that was on the ground over at Mount Tam under Douglas fir. So I'm really curious to see what kind of mycelium does that. Maybe it's something familiar, maybe it's something I've never heard of. And then here is something that might be what we're calling Cupophilus virginius in California. Though Cupophilus virginius is really from Europe and we don't have it. So Steve Ness was asking me if we had any sequences of this one. So this will be interesting to do as well. So the first thing to do is to get the mushrooms in tubes. So I've got 4 mushrooms and 4 tubes. And we have these little forceps that have been sterilized by fire. And the exact amount of mushroom that you need to put in the tube doesn't really matter very much. You can use extremely small amounts. I usually use a little more. Maybe like something about the size of a grain of rice. This is like half the size of a grain of rice. If you use more mushroom, you just need more of the extraction solution. And you can use any part of the mushroom. So now this one is dirty. We don't want to cross-contaminate, so I'll leave it back on the table so I don't accidentally reuse it. And we'll burn that with fire later to clean it again. And so here's a pipette. A good pipette. This one. So I'll get the extraction solution, and this is an extraction solution we make ourselves. It's a mixture of sodium hydroxide, potassium chloride, tris, and EPA. And so I'll set this pipette to some arbitrary number. It doesn't really matter how much, but there's not a whole lot of mushroom in here. So I think maybe 40 microliters sounds reasonable. So there's 40 microliters. Now I'll grind this up. Stephen Russell says if you grind it, you get twice as much DNA in solution as if you don't grind it. Now we have a green solution. Let's start tonguing here. And it's really important to remember which mushroom to put in what tube. So I would write the date. 312, X1, so tube number 1. And for tube 2, I'll do this Gluteinoglossum. So I can save a little bit of plastic by not switching tips here because I never touched the tip down into the mushroom, so it's still clean. And it's a fresh forceps, so let's grab a little bit of this. And these pestles are supposed to be disposable, but they're kind of expensive, so we just soak them in bleach and then autoclave them, and then you can reuse them just fine. So that's tube 2. For tube 3, try this mysterious mycotic spore mat. I'll try to get as much of the actual mycelium and as little of the dirt as possible, but it's really just a thin film of mycelium on top of the dirt, so there will be some dirt in there. And that might be okay or it might not be. And we'll find out when we run the electrophoresis or we get the sequence back. Let's try to scrape up as pure of the mycelium as possible here. Make our chances as good as possible, getting a good sequence out of this. Now, number 4 will be this Carpophilus virginius. It's definitely not going to be Virginius, so it'll be real interesting to see what it really is. So I'll just grab some of the gills. Todd Osmondson says maybe there's more DNA in the humidifore where it's making spores, so I usually try to grab some of the gills. So the next step is to heat these up to almost boiling, 95 Celsius, for 10 minutes. So we'll put those in the heat block.
And again, for you folks tuning in on YouTube, I'm doing this with my Chromebook. So if you want a better, closer up experience, I would suggest switching to Alan Rockefeller's Facebook page, where he's doing a live stream from his phone, and you'll get a little bit more close up. Now, after all is said and done, I'm going to edit together a video. I've got this on my DSLR as well, and so I'll put a full YouTube video up on the channel that'll be edited and hopefully get a little bit more detail going for you.
So here's a heat block, and this is at 95 Celsius, so almost boiling. And you can skip this step, but it works about 20% less often if you don't heat it up.
And if you're at home, you can just heat up a pot of water to 95, so you don't need some fancy equipment. You can totally do this.
Oh, sometimes they explode like that.
Yeah, they're getting excited.
That's the problem with the pot of water is you got to tape them shut with electrical tape, so if they pop open, they don't just release all the mushroom into the water. But sometimes I've used to use like boiling water, and that works fine. So the next step in this process is PCR. And so PCR is where we actually copy the DNA. So while that is cooking there, I will set the PCR up. And PCR uses the master mix on the forward primer and the reverse primer. So the master mix has the blank DNA of DNTPs, and it has the Taq polymerase enzyme, which does the actual copying of the DNA, and some other preservatives and magnesium chloride. And then here is a forward primer. This is ITS1F. So this is a little single-stranded piece of DNA that attaches to a conserved region of DNA, of the genome, in order to tell the Taq enzyme where to start copying. And then here's the reverse primer. So this tells the attacker where to stop copying. So between where the forward primer attaches and where the reverse primer attaches, there's highly variable regions. Each mushroom has a different barcode in that region, so that's the region that we're studying. So we select the part of the DNA that we're studying based on which primers we use. And in order to figure out how much to put in, I did a little bit of math. So we're doing 4 PCR reactions, so I do math for 5 just to make sure there's enough liquid to go into the tubes. And each PCR reaction is 25 microliters. So 25 times 5 is 125, is how much total that I will mix up. And then the master mix is 2 times concentrate. So dividing that by 2 would do 62.5 of the master mix. And then the other thing we add is water. So we take the total reaction volume minus the master mix, minus the forward primer, minus the reverse primer, minus the templates, and that gives us 42.5 of pure water that we'll put in.
All this information, and I'll write about the procedure. I'll make sure to get that out to you as well. And I get a message saying the Facebook video isn't working.
Not working. Yeah, it just says starting my video. And it never started. That has not happened before. Try this again. Yep, it's not starting.
Pardon us for a second. We got some technical problems. But yeah, as I said, in the meanwhile, I'm going to be posting a edited video of all this to YouTube so that you can see it all and copy down everything you need to find out. And then I'm going to write up a procedure for all this and get you some links to the materials that you need from the Odin, the-odin.com, and that's who supplied us with all the reagents here. And they also have all the equipment that you would need to carry out this reaction at home. The primers that we're using are ITS 1 and 4. Those were developed by the Bruns Lab at UC Berkeley by a gentleman named Tom Bruns, who runs that lab now. And the ITS stands for Internal Transcribed Spacer. Oh, check that out. It's getting real psychedelic in here. Anyway, it's a section of DNA that's hidden within the DNA that codes for the small subunit of the ribosome. So it's basically a protein factory, and there's a piece of that DNA that codes for a piece of the factory. And in between it, there's these areas that have DNA that's not really used for anything. They're called internal transcribed spacers. So they don't need to be conserved very well by the organism. So as it goes through many different mating cycles and cellular division, if there's a mutation in that region, in that internal transcribed spacer, the mushroom's not going to really care as much to get rid of it. Whereas if there's a mutation in the actual part of that gene that codes for the ribosome, that means that mushroom may not be able to produce proteins properly and would never be able to survive. So that mutation is much less likely to make it through to the next generation. So because that area called the internal transcribed spacer has can be replicated without including those mutations without damaging the mushroom, you often see a lot of variability. So you can tell really well the difference between two species because there will be a predictable rate of mutation in that region. So you can feed all this data that you get into an algorithm, and it will be able to kind of tell you how different two different mushrooms are. Let's say you have two mushrooms that look very similar. One's found in Europe, one's found in the United States. They're both found in different habitats, but they've been classified as the same mushroom for hundreds of years. You think they're different. This is one great way to actually prove that, because you can show that there's been enough change over time between these two specimens to count them as different species. And you can use that as evidence in addition to a different habitat and maybe different morphological features to do that. So I think Alan is getting the Facebook video back up. Apologies on that, folks. Technology, it's great when it works.
And do you think we should start over when if this works?
Sure. Yeah, we could.
Right?
We could do a quick start over. We'll have to.
We'll have this. So this time we won't have to wait for these anymore. I can just do this part, and then we'll have these ready to go directly to PCR.
Great. That way, the folks of you who want to tune in on Facebook, you will get a quick rehash of what we just did. And those of you who are watching on YouTube, you're going to have to wait for this thing to be done anyway. We have to let this cook in the heat bath for 15 minutes. So now instead of listening to me just babble at you, you can watch Alan do the same thing that he just did over again, but in higher quality on Facebook. Or you can watch it again here on YouTube. Let's get this going. I'm not sure if this will work, but it's feeling fine. Okay. There we go. We get to watch Alan burn things.
So here we're burning the DNA from the mushroom off of these forceps. So that prevents cross-contamination. If you get cross-contamination, then you get two sequences on top of each other. It'll all be garbage. Start cleaning them with fire. We can keep all the samples separate.
Yeah, DNA is a really stable molecule. Like when people die in a fire and they're just a bunch of bones and they want to identify them, they can still find usable DNA. So we really have to burn these at a very high temperature to make sure that there's no more DNA on them.
So we got 4 mushrooms we're going to sequence here. This one's really cool. It's a microglossum, one of the green ones. And we're calling it microglossum verde, but there's 2 different microglossum verdes. In California, we don't know if either are the real verde, so it'll be interesting to see what this says. And then we have the glutinoglossum, which is a new genus that was just made, and it has this glutinous kind of viscid stem. And those just came from Ukiah. And then we have a mitotic spore mat, which is this mysterious white mycelium that showed up at Mount Tam under a tan oak. And then there is Cupophilus virginius, which is not really Cupophilus virginius because the real one's in Europe. And this one here will be real interesting to see what that matches. So we got 4 samples, and we'll put those into 4 tubes.
We're working out of a place called Counterculture Labs today in the Temescal district of Oakland. It's a community biohacker lab space where you can do all kinds of cool stuff. We got all this amazing equipment, a biosafety cabinet. This is something that you use in a hospital setting, but we have it here. We got thermal cyclers, all types of different reagents and chemicals and equipment. So come check us out, Counterculture Labs in the Omni Commons on Shattuck Avenue in Oakland.
So I'm going to put this microblossum into tube 1, and I just take a little bit. Doesn't really matter how much I take, or doesn't matter how much I take or from which part of the mushroom. So I just grab a little bit at the top. It's a little smaller than a grain of rice. I'll put that in tube number 1. Now in tube number 2, I'll put a little bit of this glutiminoglossum. And in tube number 3, I will put a little bit of this mitotic spore mat. In tube number 4, a little bit of this complex. And here we have some extraction solution. So this is made with sodium hydroxide, calcium chloride, tris, and EDTA. And so I'll just put a little bit of that into each of these tubes. This is set, we'll set this to 50 microliters, so it's a 20th of a milliliter. About 1 drop. And now I'll grind these up.
These pestles you can get on eBay. They fit really nicely in these tubes. And to clean them, you soak them in bleach and then autoclave them. The bleach will break down any DNA on them. They look like up close.
So these 4 tubes go into the heat block, and this is at 95 degrees. And so they stay here for 10 minutes, but we don't actually have to wait for 10 minutes because I have some that I put in 10 minutes ago. So now to finish this DNA extract, I add an equal amount of dilution solution as I add the extraction solution. So dilution solution is 3% BSA.
Bovine serum albumin. It's a blood protein that it helps to keep some of the other components in those mushrooms out of the reaction. And it'll bind to some of the enzymes and proteins there, and that way when we're doing the PCR reaction, they won't get in the way. Sound you were hearing was just some of the tubes popping open in the heat block. I'm going to go shut them again. Perfectly normal. You could tape them shut if you wanted.
So now we have these 4 tubes and I've added the dilution solution. And we can shake them up a little bit.
Automatic shaker thing, vortex mixer.
And now they could be done, but since we have a centrifuge here, we will spin them for a little bit. And what that'll do is just move all the gunk into the bottom of the tube. So when I pipette the DNA extract out, my tube doesn't get clogged and keeps pieces out of the PCR reaction, out of the capillary tube and the sequencer. So we'll tell it 14,000 RPM for 3 minutes.
All right.
So now it's time to set up the PCR reaction. And so to do this, I did some math here so we can figure out how much to add of everything. So we're doing 4 PCR reactions. So I'll do our math for 5 just to make sure that we have enough liquid to go into all the tubes. So PCR reaction will be 25 microliters. So 25 times 5 reactions is 125 microliters total reaction volume. So that's about 4 or 5 drops. And then the master mix is 2 times concentrated. So we'll take the total reaction volume divided by 2. So 62.5 microliters of the master mix. And then to calculate how much water we add, it's the total reaction volume minus the master mix, minus the forward primer, minus the reverse primer, minus the template. So it's 42.5 of the water.
And again, all the reagents today were donated by theodin.com, the-odin.com. It's a company that supplies biohackers with all the things that they need. You can do all kinds of crazy shit there, like genetically engineering yeast to turn fluorescent, all kinds of fun things, but they also have everything you would need to do this at home.
So here is 42.5 microliters of this super pure water. When I was at Soma Camp, I did a DNA sequencing demo and I forgot super pure water, so I used tap water instead, and it worked just fine. But since we have this nuclease-free water, we might as well use it.
Yeah, in the past I've used distilled water, tap water. Seems to not present a problem, but if you have the best, why not use the best?
So now I'll add primers. So we're doing this for five reactions. So I'll set this pipette. This is a P10, so it's 10 microliter pipette. I'll set this for 5 microliters.
You get the pipettes and the pipette tips at theodin.com as well.
So here's the ITS-1F forward primer. And that's also from the Odin. And this is a 10 micromolar concentration. So it means we have to use half a microliter per reaction. I'll use twice as much as we're supposed to use. It's easier to pipette larger volumes and it's still fine. But ITS-1F is cool because it's the F stands for fungi. So this completely ignores bacterial or any other DNA that's not fungal. So here's 5 microliters of this primer. And our reverse primer is ITS-4. The forward primer tells the PCR reaction where to start copying the DNA, and the reverse primer tells it where to stop copying the DNA. And in between where the forward primer and the reverse primer attach, there's about 650 nucleotides, and many of those are highly variable. So that's what we're going for, is these variable parts of the genome. So I'll add the master mix last, because once we add the master mix, it will start reacting and doing bad things. So 62.5 microliters of the master mix. So now pipe that up and down a bit to get it all well mixed. So we have the master mix, forward primer, reverse primer, and the water. And now this is ready to go. The only thing that's missing is the mushroom DNA.
Like it's freezing up a little bit.
Yeah, it's always better just to record it first instead of streaming. Hopefully it works somewhat. So now these have been spun down and not working. You can see all the gunk at the bottom of the tubes. DNA is in solution, so no matter how much you spin it, the DNA will not get pelleted along with the rest of the stuff.
Just trying to reconnect.
Reconnect. It's just not working, huh? Maybe we should turn off the Wi-Fi and just use my cell phone. You think it's not working at all here?
Hard to say. These people are still like waving at you and shit.
Okay, well, we could check on. We watch the stream on. It looks like it's working, but I'm kind of like watching what happened in the past. But oh nope, there's something happened. All right, we will like.
See if we can restart this off. For those of you who are watching on Facebook, we just have to restart the stream. You know, technology is great when it works.
Well, it gave me the option to restart it, so I'll try that. It'd be better if there was only one video.
Again, we're going to paste together a edited version that's going to end up on YouTube, and I'm sure we'll get a link on that on Alan's Facebook page to that. But just bear with us a second.
I'll just turn off the Wi-Fi, so I have to use my cell phone connection.
All right, thanks for sticking with us, folks. It looks like it's back up and running. So, without further ado,
Okay, so we're ready to start the PCR. So the next thing to do is to put this this solution. So this is the master mix, forward primer, reverse primer, and water. And we'll put it into the 4 PCR tubes. So I'll put 24 microliters of this in each of these. At this point, they usually just kind of hold it up to the light to make sure that they all have the same level in there to make sure I didn't skip any or hit any twice.
It's important when doing this to have these little systems to make sure that your workflow is consistent and you don't make mistakes.
So here's a 2 microliter pipette. So this is set to 2. So you probably can't even see in there. It's about a millimeter of liquid. So it's a huge, or a really tiny amount of liquid that goes in here. I'll give it one more shake for good luck, and now it's time to go into the thermocycler. Here is a thermocycler here. All this machine does is heats up and cools down this aluminum block. We'll create a new program. And what we'll do is 94 degrees for 3 minutes. And that splits the DNA double helix into 2 separate strands. And then we'll repeat what's between these 2 bars 35 times. So we're splitting it into 2 strands at 94. And then we'll go down to 54 degrees. And at that temperature, the primers will attach. And then we'll go up to 72 degrees. And at this temperature, the actual copying will happen. And probably 40 seconds is enough for that. And then we'll tell it to start. We have 26 microliter reaction volume. And now we just wait a couple hours.
Of course, we're not going to make you sit here and watch us twiddle our thumbs for a couple of hours, although it might be exciting. Now, we've taken the cooking show approach, and we already have some product that's been through the PCR reaction, so we can go ahead and show you the next steps.
The next step is gel electrophoresis. And what that does is it separates all of the DNA that's in the PCR product, separates it by size. So it's going to test our PCR so we know if it worked or not. And if it did work, then we know that it's worth spending the money to get it sequenced. If that phone runs low on battery, here's the charger for it.
Great.
So here's a gel box here. Are these the ones that we're going to use, Hart?
Those are just some empty tubes. I can grab you the ones that are already...
Yeah. And the gel, I suppose. The blue ones, okay.
So I'll put instructions on how to make one of these agarose gels that we have here. I'll put that in the procedure. And what the gel is it's a polysaccharide. It's called agarose. It's basically forms a kind of skeleton inside of a liquid. It's basically like Jello. But as the DNA gets moved through it, that structure that's formed prevents larger molecules from moving through, whereas smaller ones can move through really easily. So it'll separate things out based on their size.
So what we have to do first is mix the PCR products with loading dye. And so here's some loading dye.
And you made that yourself, right?
No, I think this loading dye was some commercial loading dye.
If you wanted to make that, all you'd need is glycerin and some food coloring.
Yep. So I'm going to put 8 drops of this loading dye out on the parafilm.
Just a sheet of parafilm. You can use a sheet of wax paper. Parafilm is just fancy wax paper that stretches.
And now what we have to do is mix our PCR products. And do you still need to sequence these, or should I?
I don't need to sequence those.
Don't need to sequence them. Cool. So I'll set this to maybe 7 microliters. And at this point, it's real important to switch pipette tips so we don't contaminate from one to the other. We'll do 7 after.
7, yep.
Cool. So we'll put a ladder in for the 8th then. And let's see, here's a gel.
So this is agarose. It has the buffer that we're going to use. That's the electrolyte solution in the electrophoresis box. So buffer meaning that it holds a constant pH, and electrolyte meaning that it allows electricity to flow through a liquid medium. So that's in that gel, along with a compound called gel green. And the gel green attaches itself to DNA and then fluoresces under blue or fluorescent light. In our case, we're going to use blue light because it's much safer. If we're using fluorescent light, we'd have to use something that's 260 or 360 nanometers, something really not so good to look at. And that's going to allow us to visualize the DNA in the gel, because it'll actually stick to it.
Well, gel green's a little bit poisonous, so I just washed my hands so we don't get it all over the pipettes.
So this is the part that requires a bit of dexterity. It's going to be hard to see this on the camera, but what he's doing is pipetting the droplets of DNA that's been mixed with a loading dye. He's mixing those in the pipette and dropping them into these little wells that are cut into the gel. And the loading dye contains something like glycerin, which is heavy, and it's going to actually weigh down that mixture inside that well, inside of the gel, so it doesn't just float out into the electrolyte solution. So in this circumstance, the DNA is negatively charged, and so when we run a DC current through this electrolyte, the DNA is going to want to go to the positive electrode, because negative is, gets attracted to the positive. And it's going to happen inside of that gel, and that gel has a structure in it that will prevent larger molecules from moving as fast as the smaller molecules. And actually, we've just copied one little region of DNA, so we're really only looking to see one band in that gel. But what we're going to go ahead and do is put something called a DNA ladder into the gel, and that's a mixture of a bunch of different sized pieces of DNA. And with that ladder, you can actually measure the piece that you have in the gel. It's going to show you what looks like a ladder of all the different sized pieces.
Is the ladder in here?
The ladder should be in there, or...
Well, I think we have it out.
Yeah, it's in the other primer block. See, Alan's got quite a collection of primers here. You can only find the ITS ones on theodin.com. That's the-odin.com, but you can custom order primers from a company called IDT. GeneWiz, which will be doing our sequencing, they also design primers.
All right, so now it's time to run some electricity through this. So this is where we tell it how many volts. I think a good number of volts is 140. So we could do 200, 150. That'll work. The higher voltage that we choose, the faster it'll go. So I just did 160. And then we can tell it how much time. This way it won't accidentally run off the gel. So I think we have a lot of room here. So I'll do 16 minutes.
Now just keep in mind, folks, if you're doing this at home, that 160 volts, not something you want to stick your fingers in there. Don't lick it. It is high voltage there.
So now it's putting positive here and negative is here. So this will pull the DNA towards the positive and through the gel.
You could certainly buy this equipment on theodin.com, or you can look up a YouTube video on how to build it yourself. You can actually make this power supply out of a stack of 9-volt batteries, and you can use a Tupperware container as your little gel box.
Okay, well this will take 15 more minutes. So while this is running, let's do some sequence analysis.
Frozen up for a second.
Oh yeah. No. Hopefully it sort of works enough. So here's a bunch of the AB1s that I have here. Trying to reconnect. Maybe it'll work. So I'm gonna let you do that second. Hold that second type. So here's one from a workshop that I did in Eugene, and so I'll use a program called Snap Gene Viewer. And we'll load in this AB1 file, and AB1s are chromatograms, so we get a graph with the different different bases. And this one did not come out well. You see, you have a real choppy graph. You have a lot of garbage in here. There's probably enough information in there to actually identify the mushroom, but it's a pretty noisy sequence. Let's try a different one.
Looks like it's being a little tricky still.
Yeah. Whatever. We tried. But then it quickly it starts to get. So looks like the garbage stops about there. So I'll chop off the beginning, first 28 bases here. And this looks pretty reasonable here. And the data continues to look good until we have this N here. And if we look a little bit more closely, what we can see is that the reds are T's and the blues are C's, and we have a T and a C in exactly the same spot. Or we could just use a nucleic acid ambiguity code. But looks like the all the way to the end of the sequence is good. So I will copy that onto the clipboard. And now we'll go to NCBI BLAST. So we hit nucleotide BLAST, and this will compare our DNA with all of the other DNA in GenBank. So it's really easy. We just paste it into the query box. All these settings, the default looks fine. And then we click BLAST. And
Can't continue live video.
Can't.
Pardon just a second, folks.
Well, I guess it wasn't the wireless that was causing us problems.
Well, with all the crap going on in the world, I'm sure there's plenty of people just hogging the social media pipeline.
All right, this is working. So here's our BLAST results. And first we see all these pink bars here, and we can see the bars are going almost all the way across the screen, which means that we get good query coverage, which means that we'll get relatively reliable results. And all of the top BLAST matches are Spirasus radicata. So this is Sidney Obersingleton's Petri dish culture. And so he cloned the Spirasus onto agar, but he wasn't totally sure that what was growing really was Spirasus. So we sequenced it, and it turns out that it really was Spirasus growing. So if we click on this, we can see the alignment, and our sequence matches 596 out of 597 with the other sequence here, and the only difference is this N here. And so this reference sequence has a T there. And so what we should do is take a look at some of the other reference sequences and see if they're all like that. So there's a T in this one, a C in this one, a Y in this one. So that means that we definitely have a double base there. So it's no accident that we have a T and a C at the same spot. So the best way to handle this would be to put a Y here, and Y is nucleic acid ambiguity code, which means that it's either T or a C, because there's multiple copies of the ITS in the genome, and some of the copies have a T there, some of the copies have a C there. So being able to put a Y here is good, because that lets us know that there is that difference, and that's something that you'll see in the sequences. So with that change, that means that we're matching 100% with all these other Spiraceus radicatus sequences in GenBank. And looks like there's only one species in GenBank going under the name Spiraceus radicata, so not a species complex. Um, the other Spiraceus species are further apart.
Yeah, so that percent identity, that's going to help you figure out, again, if it's a single species or if it's a complex. But how many base pairs different would you be looking for, or what percentage different would you be looking for if it was in a complex?
There's no set number. It just depends on how you like to define the word complex.
There you go.
So we got a whole bunch of different sequences we could look at here. One of them is a chromosera. Maybe that'd be interesting. So this is a 368-868. So if we want to see what we're doing here, we go Mushroom Observer 368-868.
And what's that little terminal you were using on your computer there, Alan?
This is called Terminal. It's a bash shell.
So it's a different way to navigate your computer?
Mm-hmm.
Cool.
Well, this is Linux, so that's pretty much how I do everything is through the terminal. This one is called Chromosera cyanophila, and it's from the Crystal Basin, Icehouse Reservoir, El Dorado National Forest. So that's up in the mountains of California. And so looking at the sequence, looks like it's got garbage at the beginning and ends, but the middle is pretty good. So I will chop it there, and this looks pretty reasonable here. Nice even peaks, even spaces between them. And then it turns into garbage around here. So I'll delete that. And now we have just our good data. Copy it to the clipboard.
So this is Blast Search. It's basically searches a database of all the DNA that's been entered into it, of every organism, and tries to match it against something. Of course, people have to actually add that to that database, and that's called GenBank, and you can do that yourself with your own sequences. Alan probably has a couple hundred up on there.
500.
500, there you go.
So here's our results. And you can see the query coverage is pretty good until you get down to here. So not bad for the top results. And our top match here, Chromosera F cyanophila from 317-467. So if we look, we can see where this top match is from. This is also the El Dorado National Forest. So this is a really cool purple little mushroom. And take another look at our matches here. So if I click on the top match, we can see that 580 out of 584 are the same. And so I want to see what the differences are here. So by searching for pipe space, I can make a highlight the differences. And the first set of differences are over here. So what it'll do is just copy some of this DNA sequence right before the difference starts, put it on the clipboard, and then go to our chromatogram and search for that place. And there it is here. And what I'm seeing is that the sequence that we're searching against has some problems with it. It has some problem in the read, but our sequence is not having problems in the read. You can see that this is where the problem is, and it looks real nice there. So that means that we have a better sequence of this mushroom than this other reference sequence, and that's cool. And then down here, we got a T where they have a C. So let's see what's going on over there in the chromatogram. And definitely we have a T and the C in exactly the same spot. So that should be a Y, indicate that we have a heterozygous base there. And then over here, we have a C where the reference sequence has a Y. See what's going on over there. And we do not, that's not exactly what we're seeing here. We see a little bit of a G in the background, but it's a pretty strong C compared to that G. So I think we'll just have a C there. So there's really just one difference between these two. So almost certainly the same species because it's so close. And then if we look at the next one is 99.4% the same. And seeing similar stuff. So this this record says that it's from Wyoming. So it's interesting to know that this California species also occurs in Wyoming. And then if we look at the next closest one, it's down by 95.89%. So this is a different species here. And so now our next question is, where is this other species found? So we'll open up a few tabs to see where these other species are found. And you can see they have the same name. So you got a couple different species going under the same name. So that means we just uncovered evidence of a new species. This one's in Austria. So this is the European one. So this Chromosera cyanophila is named from Europe. So that means that this sequence here will be the real Chromosera cyanophila. And what we're finding in California and Wyoming is not Chromosera cyanophila. Here's one from the Czech Republic, Russia, the Netherlands, Greenland, and then here's an even further apart one, Chromosera species. These have some iNaturalist numbers. This one is 242, 223, 14. So let's see what's up with this one.
Internal storage space.
Dangerously low. Oh, and this one's out in Indiana. So that means the East Coast one and the West Coast one and the European one are different. So we have three different Chromosera cyanophilas. But I just heard the electrophoresis go off, so let's go see how our gel turned out.
We're going to have a look at the gel right now.
So you can see that it's moved about 15 millimeters to the right.
Those blue bands that you see are the dye that we added there to show us where the DNA has moved, as otherwise we wouldn't know where it went.
And you have the transilluminator in the back room.
Yep, it's going to be in the back room where it's nice and dark.
So here's a transilluminator. All this does is makes a bright blue light. So we will take our gel and put it on there. Turn off the lights. And now all we need to do to see the results is put this orange filter over it. Oh, wow, this looks really good. So out of 7 possible ones that could have worked, all 7 worked. So you can see those bands there are the PCR product. And since it's a band, we know that all of the DNA is exactly the same size. And then here's the ladder here. So we can tell it's about 600 base pairs long. So this means that these 7 PCR products are ready to be sent off for sequencing, and we'll get good results from them if we send them off.
Sweet. Well, that's pretty much the process from start to finish. We're going to be holding an in-person class on April 23rd. That's going to be I believe, the 4th Thursday in April. And hopefully things will be a little bit more calmed down by then. And anyone who's welcome can come visit us at Counterculture Labs for that class. That's in the Temescal in Oakland at the Omni Commons. And follow the YouTube channel. It's a CCL Mushrooms, CCL short for Counterculture Labs. And I'll be posting an edited version of this video, as well as some links to a written up procedure and some other information if you want to do this stuff at home on your own, or if you ever want to visit us at Counterculture Labs and learn to do it right here.
All right, if you have any questions about any of this stuff, you can message me.
Yes. All right. Thank you, folks.
All right, that worked. Sort of, maybe.